Okay, we're ready to start a new chapter. This is chapter 14 on blood. And this first slide shows a scanning electron micrograph of one of the three formed elements of blood. These are red blood cells, or erythrocytes, which, as it mentions here, blood in general, if you look at all of the blood volume in your body, makes up close to 10% of your entire body weight. So it's pretty substantial from that standpoint. And volumetrically, if I were to drain you of all your blood, we could fill up basically 2.5 of those 2-liter Diet Pepsi bottles, if you can all envision kind of what that is. So there's quite a bit of blood in our body. Here we see the three types of cells in our blood. These are sometimes referred to as formed elements, and those are indicated, of course, by the asterisks. You all have heard of red cells, white cells, and platelets. We'll be talking more about those in some detail, obviously, in this chapter. Had we had lab, one of the exercises that would have been available to you would be to have your blood spun down in a machine called a centrifuge. And what we would have done is we would have used a small little glass heparinized capillary tube, very similar to what you see here on the sketch. Heparinized means a chemical referred to as heparin has been added to the inside of this little hollow glass capillary tube, which will prevent the blood from clotting. So you basically prick your finger and you hold this capillary tube at a bit of an angle downward, and the blood is drawn in through capillary action. You fill it about 4/5 full or so, and then you cap it here with a piece of clay. So it's basically open at one end, and you put this into an instrument called a centrifuge, and you spin it for, I think it's like 5 or 6 minutes at a very high RPM, and it comes out and you see what's referred to here as a hematocrit. This is the lower portion where we have primarily red blood cells occupying about 45% of the entire blood volume, with the remaining 55% or so being the liquid plasma, which is what bathes the blood. This is the matrix of this connective tissue. Remember, blood is a connective tissue. And sandwiched between this 55% and the approximate 45% of the packed cell volume of red cells, we see this little buffy coat zone here, which of course contains the other two types of formed elements, the white blood cells and the platelets. So this gives you a really good idea of just which of the three formed elements is most abundant in our blood, and that of course is the red cells. We have many fewer white cells and platelets than we do red cells. So it's too bad we can't do that this semester in lab, because this is kind of a fun little exercise. So here we see again the three formed elements making up approximately 45% of our blood volume, give or take a percent. And then we've broken this down into various compartments, or categories I should say. Again, red blood cells occupying over 95% of the formed elements, followed by platelets, a little under 5%, and notice here 0.1% of our blood cells are white cells. I bet you were surprised at seeing that statistic. You would have expected perhaps a lot more white cells. Now certainly you would have a higher count if you were sick or had an infection, because remember, these are the cells, of course, that are fighting infection for us. The red cells primarily carry oxygen and to some extent some carbon dioxide. And what do platelets do? Well, you might remember they're involved in helping to stop bleeding. And then notice that the white blood cell category is in turn broken down into these 5 different kinds of white cells, or leukocytes. And we'll be talking about the neutrophils, which are the most abundant of the 5, and then the lymphocytes, which come in second place, and then here comes the monocytes, between 3 and 9% of circulating white cells, followed by eosinophils, and then the most I won't say rare, but the least common white cell called the basophils. 80% of our blood, in terms of volume, is the watery plasma. Remember, this is the matrix of our connective tissue. And note that the majority of the plasma, 95%, is good old H2O, followed by a variety of plasma proteins, the albumins, globulins, and fibrinogen. We'll talk more about those a little bit later as well. So there's what, 99%, with the remaining 1% sprinkled between electrolytes, which are charged ions, different waste products, nutrients and vitamins and different hormones that circulate in our bloodstream, and even gases need to be considered here. We're most familiar with oxygen and CO2, but there's even a little tiny bit of nitrogen in our blood. Not much, but a little bit. This is talking a little bit about how these formed elements, the red cells, the platelets, and the 5 types of white cells, are produced by and in our red bone marrow, which remember is in the spongy bone. And we have in our red bone marrow a type of stem cell referred to as a hematopoietic stem cell or hemocytoblast, same thing. And here's an actual photograph of a nice big hematopoietic stem cell. As you remember from, I think, chapter 3, stem cells have this very unique ability to differentiate into a number of different types of cell types. And that's indeed the case here, where we have this hemocytoblast potentially differentiating into what's referred to as a myeloid stem cell or a lymphoid stem cell. So it's sort of like a close cousin to the hematopoietic stem cell, although this guy has a bigger variety of options in terms of what it can differentiate into, right? Either one of these. And then once we look at each of these 2 types of stem cells, we can see that they in turn have the ability to differentiate into a number of different kinds of cells, depending on whether or not, excuse me, they produce various types of growth factors. I'm not going to get into a lot of detail about how they work, but as I've indicated here in the slide, some of these chemicals, these growth factors, have the ability to activate certain types of genes where they're expressed, while other genes might be turned off, which in other words means they're not expressed. So if we look at the myeloid stem cell, note what it can potentially differentiate into. It can differentiate into a number of different types of cells, which culminate in the formation of red cells or erythrocytes. Some myeloid stem cells can differentiate into these big megakaryocytes. These are really large cells. But look what they produce. Tiny little bitty platelets. I think it's kind of ironic that this huge megakaryocyte differentiates into the smallest little fragments of cells called thrombocytes. Some myeloid stem cells can also differentiate into 3 of the 5 types of white cells, the neutrophils, basophils, and eosinophils, collectively referred to as granular leukocytes or granulocytes, because in their cytoplasm, if you stain these cells with a particular type of stain called Wright stain, you can see these little granules. They're very apparent. You'll see some pictures of those a little bit later in another voiceover coming up. So they have little granules, stained granules, and that's where they get the name granulocyte. And then finally, the last type of white cell that the myeloid stem cell could in turn differentiate into is something called a monocyte. And if that monocyte leaves the bloodstream, which it sometimes can do in the case of an infection perhaps, we then refer to it as a macrophage or macrophage, some people pronounce it. So this cell, the monocyte, when it leaves the bloodstream, then is redefined, if you will, and is now called a macrophage. And this is a very highly phagocytic cell, as you can tell from the suffix of the word, phage meaning to eat, macro meaning big, and it is fairly large cell here. So it's very hungry, likes to gobble up bacteria, virally infected cells, cancer cells, and that kind of thing. And then here's our lymphoid stem cell, which notice has the potential to differentiate into the lymphocytes. And we'll talk more about T and B lymphocytes in the lymphatic chapter in a couple weeks. We wrap up the semester with that system. And yeah, again, notice that some B cells, when they leave the bloodstream and get out into the circulation, can form into something called a plasma cell. And this is going to be an important cell in the immune response because it secretes what is referred to as antibodies to help fight infection. This slide just a recent discovery that was made in the last 10 years or so that discusses formation of blood cells here in the lungs. We used to always think, of course, that most of the formed elements were formed in the red bone marrow, but we've come to discover that the lungs are an important source for blood cell production as well. I thought that was kind of interesting, so I've included that here. Make sure you check out that link. Okay, let's take a look at the red cells. Again, we looked at an earlier micrograph. This is quite similar. And what this refers to, of course, are 6 or 7 individual erythrocytes or red cells. Notice that they're biconcave, sort of shallower or thinner in the middle than they are around the edges. In adult males, we typically like to see RBC counts of somewhere between 4.7 and 6.1 million cells per microliter or cubic millimeter. Just a little bit lower than that, 4.2 to 5.4. And the average life expectancy of a red blood cell is about 4 months, and then they're recycled, which we'll talk a little bit more about in a few minutes. So they're roaming around the blood for about 4 months, helping to transport primarily oxygen and some CO2 from our lungs to our tissues, and then from the tissues back to the lungs. And this gets at an important function, of course, with regard to oxygen carrying capacity, because it is this vehicle, this cell, this red blood cell, which notice lacks a nucleus. It's one of the very few cells in our bodies that doesn't have a nucleus. It loses it during that differentiation process that we looked at a few slides prior. If you go back and look at that slide, you'll see that it loses the nucleus as it develops from that myeloid stem cell. So when we talk about the oxygen carrying capacity of the blood, we are interested in oftentimes the number of red blood cells, because the greater the number of red cells, the greater our oxygen carrying capacity, that we can deliver that O2 to our metabolism. Inside your red blood cells, again, no nucleus, but a lot of a particular protein called hemoglobin. About 1/3 of the volume inside each red cell is made up of hemoglobin. Note also its flexibility, as it is noted here in the slide, because when these cells move through capillary beds, if you recall from the respiratory system, they do so in single file, and they literally do squeeze through those small little capillaries. And what happens, remember, at the capillaries? We have that exchange, don't we, of CO2 for oxygen, as it pertains, of course, to the respiratory system. We talked about a little of that earlier. And so that diffusion process occurs most optimally at those capillary beds. Getting back to the hemoglobin, of which there are millions and millions of molecules of this in each one of your red blood cells, we see this sort of model here depicting the 4 different polypeptide chains. They're denoted by various hues of purple here. And in the center each of those polypeptides is what looks like a little flying saucer almost. This is referred to as a heme group, in the very center of which is iron. Maybe you associate adequate iron in the diet with red cell function. And indeed, that's certainly the case. We'll talk about the importance of iron in just a couple seconds. So we've got millions of these molecules of hemoglobin in each red blood cell, and this molecule is going to be instrumental in helping us transport a lot of oxygen. We've talked about negative feedback quite a bit in the last few months. This is another example of maintenance of homeostasis as it pertains to oxygen carrying capacity of our blood. It's been shown that if your blood oxygen level falls, which could be a result, excuse me, a result of going up in elevation, or losing blood, or having some sort of a chronic lung disorder like emphysema, perhaps, that there are special receptors in our liver and in our kidney. And the double asterisk here in kidney simply is indicating that the kidney plays perhaps a little more important role even than the liver in detecting that drop in blood oxygen levels. But in either case, these two organs will detect that drop in blood oxygen, and they will produce a hormone called erythropoietin, or EPO for short. That EPO is released from these two organs. It gets put into the bloodstream. It circulates throughout our entire body. But as you may know, hormones affect particular target tissues and the cells of those tissues. And in the case of EPO, the target tissue is the red bone marrow. It's those hematopoietic stem cells that are stimulated to differentiate into more red blood cells. So as a result of the erythropoietin, the red bone marrow produces more red blood cells. That in turn increases the oxygen carrying capacity of the body. And as that number of red cells goes up and as that capacity increases, this results in a reduction in the production of erythropoietin by these two organs. It's classic negative feedback. So make sure you check this out. Very important to understand. Let's talk a little bit about some dietary factors that influence RBC production as well. And this is going to talk a little bit about a couple different substances, vitamin B12, iron, and something called folic acid. And you'll notice that all of these three substances are absorbed by our small intestine, and two of the three influence the formation of DNA within the hematopoietic stem cells as those stem cells differentiate, as they divide, right, into ultimately the various types of formed elements. They need to have adequate vitamin B12 and adequate folic acid to allow for DNA replication. We talked about that back many chapters ago. So without adequate vitamin B12 and without adequate folic acid, this can directly impact the formation of blood cells because the hematopoietic stem cells cannot replicate their DNA. Iron, as we said a few moments ago, was part of that heme group. It's part of the hemoglobin and also absorbed by the small intestine. This particular compound, or really it's an element, is absorbed by our body as we eat certain foods like spinach, and I think egg yolk is high in iron, and various other types, cod liver oil, I think. This certainly helps in the formation of the hemoglobin. Specifically in the center of each of those little flying saucers is that iron where the oxygen is going to bind. And as we'll be also talking about, not only do we absorb iron via the foods we eat, but as we recycle those worn out red blood cells after 4 months, we said they are recycled, they die, right? They're recycled by the body, and the iron in the hemoglobin is reused to make more hemoglobin. So that's kind of interesting. We have all heard the term anemia, and there are different kinds of disorders that are of this nature, but regardless of the type of anemia, we typically talk about either a reduction in the number of red blood cells or a reduction in the amount of hemoglobin. So in other words, what does anemia do to the oxygen carrying capacity of the body? Well, it reduces it, doesn't it? On the top photo here, we see some normal red blood cells. Again, notice their nice circular biconcave shape. The reason that we see these little white zones in the center of each cell is simply because they're thinner. Remember, in the middle, and light passes through them much more easily than around the periphery, which of course is shown here as a darker red. I want you to contrast this normal array of red cells to the bottom photograph. And as you compare the two, of course, you see the size is about the same, but you see more white here in the center of each cell. What does that tell you? Well, that tells you it's much, much thinner, right? And the reason for that is because these particular red blood cells have less hemoglobin in them. The proportion of hemoglobin in the volume of that cell is much reduced. And if you have less hemoglobin, what do you know about the oxygen carrying capacity of somebody with this kind of blood? It's going to be reduced, isn't it? And so this is due to not having sufficient iron in the diet. And excuse me, as a result, that deficiency of iron is directly impacting in a negative way the ability of the body to get perhaps adequate oxygen to the tissues. Here's another photograph that's showing what's called hemolytic anemia. Remember, hemolysis is rupturing of red cells. We saw that when we added hypotonic solutions to red cells a couple weeks ago. Excuse me. This isn't due to being placed into a hypotonic solution, but is often due to cells being subjected to various nasty chemicals. When cells are invaded by certain kinds of parasites, they can also rupture, which is the case in malaria. But typically, hemolytic anemia, if you're popping those cells, you obviously are significantly reducing the RBC numbers and the oxygen carrier capacity of the bloodstream. This table shows, of course, several different types of anemia, their cause, and how that can impact the body. Here's sickle cell disease, sickle cell anemia. We talked about this back in one of the earlier chapters. We said that the misshapen cell here was due to a single different base added within the gene that results in the abnormal shape of the cell. Let's talk a little bit about the recycling of the red blood cell. Remember, we said after 4 months the red cell is recycled by the body. A lot of the iron in the hemoglobin is used again to form new hemoglobin as more red cells are produced. So this is taking you through the 6 different steps in the life of a red blood cell. I'll let you read through that. And let's draw our attention to the liver here, where those red cells are going to be actually broken down. Back to our diagram of the hemoglobin. Notice in the center of each of the polypeptides is the heme group. It's this flying saucer. And in the middle of the flying saucer is this sort of round structure. That's iron. And that's where the oxygen binds to the iron of the into the heme group of the hemoglobin. So there's 4 oxygens that can bind to each hemoglobin molecule. So we're going to zoom in on one of these heme groups, one of these little flying saucer looking things. And we're going to look at the chemistry. We're looking down from above onto the flying saucer. Here's the iron in the very center. That's where the aliens live. And here's the other part of the flying saucer. This is the various 4 rings that can hold that iron in the middle. When our hemoglobin is degraded by the liver after 4 months, the red cell is phagocytized by the macrophages and broken down, and then the hemoglobin is chemically degraded as well. So we're taking that 4-ring structure that we saw in the previous slide, and we're snipping a couple bonds. We're breaking some bonds. We're pulling the iron out of the center, and there's those 4 rings, still connected together, but the iron's, of course, detached. And this particular compound here, this 4-ringed compound, is referred to as biliverdin. It's kind of a funny name. And if you were to see biliverdin, it would have a greenish color to it, green pigment. The liver, again, produces this. as it initially degrades the hemoglobin. That iron, notice, as I said a moment ago, can be recycled. Isn't that interesting? Or if it's not used right away to make more hemoglobin in the red bone marrow, it can be stored. Your liver can store iron. So a liver is very, very rich in iron because of its storage capability. Now this biliverdin that we were talking about, this greenish pigment that was produced initially from hemoglobin degradation by the liver cells, eventually this stuff gets converted by the compound that you probably have heard of called bilirubin, which is an orange pigment. So we've rearranged some of the elements here in those four ring structures. Again, think of it as a close cousin to biliverdin, but this is bilirubin. Bilirubin. These two byproducts of hemoglobin degradation, biliverdin and bilirubin, as it mentions here in the box, are excreted in the bile. You've heard of bile before probably. Made in the liver, stored in your gallbladder. When you eat foods rich in fat, and that food moves from the stomach into the small intestine, there's a duct, cystic duct, that leads from the gallbladder, and it dumps into the first segment of the small intestine called the duodenum. It's the first little 18 inches of your small intestine, just after the stomach connects. So when foods rich in fat are eaten, the body responds by producing increased amounts of bile pigments. And the bile helps to degrade or emulsify fats. These aren't enzymes, but they're materials that help to break down the bigger fat droplets into smaller ones, which are then acted upon by various lipid digesting enzymes. So this is just a review of the various things we were talking about leading up to the recycling of the iron. This table likewise just sort of reviews the major steps.